And in particular, we look at the immune system and how these cancers are actually extremely immune cold, so they don't have any infiltration of T cells or NK cells that are able to fight the cancer. So we're looking at using CRISPR-Cas9 as a screening tool to really conduct these large-scale whole genome or boutique libraries with CRISPR screening to look for genes that are going to increase the response of these tumours to T cell killing and thereby find new targets for immunotherapy. My name is Dr. Mara Zeissig. I'm a lab head here at the South Australian Immunogenomics Cancer Institute at the University of Adelaide, and I run a research lab, the T cell immunotherapy lab. So I did my undergraduate degree here at the University of Adelaide, and then I actually really found research really interesting, and so I wanted to pursue a PhD. And at that point, the university had the option of entering into a master's degree and then converting that into a PhD, so which allowed me to essentially fast track the time that it would take me from undergraduate to graduating with a PhD. So I did that. So I didn't do honours. I just went straight into the PhD, which luckily worked out. So I think that was a really good experience for me. It really threw me into the deep end really quickly in terms of research. So when I finished the PhD, I then moved to Melbourne. And this was to pursue my first postdoc. And I think I moved 2 weeks before the very first lockdown in 2020. So moving to a new city then was perfect timing, especially Melbourne in 2020. But luckily it was still a very productive time research-wise. So I completed my first postdoc at Monash University, and then I moved to my second postdoc at the Walter and Eliza Hall Institute, or the WEHI, in Melbourne CBD. So I think the decision to move to Melbourne was really a critical one in terms of my career. So that really, I changed from doing blood cancer research here in Adelaide for my PhD to doing what I do now, which is solid tumour immunotherapy. So it was a complete career shift there. But I think it was extremely beneficial, because what then happened is I was recruited at the end of 2024 to move back to Adelaide and start my own research lab on solid tumour immunotherapy. My lab focuses on pancreatic cancers and lung cancers, and both of these cancers are really quite horrible. So they have very low survival rates and very poor responses to treatment. And really, within the first sort of 5 years post-diagnosis, less than 20% of these patients are still alive, which is extremely sad. So these were cancers that I found very interesting, and where we can really make a huge impact in terms of the translational research. So this is where the focus is for my lab, and in particular we look at the immune system and how these cancers are actually extremely immune cold, so they don't have any infiltration of T cells or NK cells that are able to fight the cancer. So we're looking at using CRISPR-Cas9 as a screening tool to really conduct these large-scale whole genome or boutique libraries with CRISPR screening to look for genes that are going to increase the response of these tumours to T cell killing and thereby find new targets for immunotherapy. And so this is really going to allow us to develop new immunotherapy combinations that are actually going to be successful in these really immune cold and highly aggressive solid cancers. And so that is really sort of the main focus of the lab. But we also focus on targeted therapies. So there are some, especially for example, the KRAS inhibitors. So most lung and pancreatic cancers are driven by KRAS. And this is an oncogene that is, it leads to high proliferation in the tumour cells. And it also leads to a highly immunosuppressive microenvironment. And so the KRAS inhibitors have been shown to remodel the microenvironment and make it more permissible to immunotherapy. So this is something that we're also interested in, and seeing how the immune system is altered with targeted therapies such as the KRAS inhibitors. SAiGENCI is located within a biomedical precinct in Adelaide. So we are right next door to the Royal Adelaide Hospital. And this allows us to work quite closely with clinicians to access patient samples. And this is critical because then we can look at, for example, in terms of the immunotherapy research that I do, is whether a gene that we found is going to increase response to immunotherapy if we target it, whether that is actually expressed in patients that don't respond or do respond to immunotherapy. So it helps us to validate our research that we find in our preclinical models within clinical samples. So it's a really critical part of our research. We also work quite closely with researchers in Melbourne who are also working on lung and pancreatic cancers to really create a sort of environment of quite collaborative research to drive the projects forward, as opposed to creating sort of silos where every lab works independently. When I was doing my postdoc in Melbourne, this was funded through the Victorian government through an early career research fellowship. And I was also funded through Cure Cancer to look at KRAS inhibitors in lung cancer. And both of these grants were really extremely important at the early career phase that I was in when I received this funding, and allowed me to work for 3 years in Melbourne running these research projects. I've also recently been funded through Tour de Cure to look at KRAS inhibitors in pancreatic cancers for the new lab here at SAiGENCI. I think it's really about knowing that what we do in the lab can actually make a difference to patients. So one of the reasons why I've focused on lung and pancreatic cancers is because we see such detrimental effects in the clinic. And this is really somewhere where any small change that we can make, or any progress forward in terms of a new therapy, is actually going to be the most beneficial. So this is what really drives my work. I'm also particularly passionate about looking at immunotherapies, because if we think about it, our immune system is really quite fantastic at clearing viruses or bacteria or any sort of infection that we encounter, and yet it is really difficult for the body to clear a cancer cell because it's out one of our own cells that's gone rogue. So the fact that we can harness the immune system to be able to help it clear cancer is really, really fascinating to me, because as opposed to something like chemotherapy where there'll be some cancer cells remaining alive, your immune system is not going to miss a single cell if we get it right. So it's really, I think, where the future of cancer therapy is, and where we can potentially find cures for this disease. I'm actually expecting my first baby in the next few weeks, so that's going to be completely turned my whole world upside down. So that is something that is really exciting, and it's coming out really soon. I also really enjoy being outdoors, so we've just purchased a new property in the Adelaide Hills, so it's extremely big and we have no idea what to do with it. So, but it's really exciting to have, because I was raised on a farm, so having animals around you and trees is really my happy place. So now between expanding family and new property, there's a lot of things to look forward to.